The superior colliculus, Latin, upper hill, is a paired structure of the mammalian midbrain. In other vertebrates the homologous structure is known as the optic tectum or simply tectum. The adjective form tectile is commonly used for mammals as well as other vertebrates. The superior colliculus/optic tectum forms a major component of the midbrain. It is a layered structure, with a number of layers that varies by species. The layers can be grouped into the superficial layers, stratum opticum and above, and the deeper layers, the remaining layers. Neurons in the superficial layers receive direct input from the retina and respond almost exclusively to visual stimuli. Many neurons in the deeper layers also respond to other modalities, and some respond to stimuli in multiple modalities. The deeper layers also contain a population of motor-related neurons, capable of activating eye movements as well as other responses. The general function of the tactile system is to direct behavioral responses toward specific points in egocentric, body-centered, space. Each layer contains a topographic map of the surrounding world in retinotopic coordinates, and activation of neurons at a particular point in the map evokes a response directed toward the corresponding point in space. In primates, the superior colliculus has been studied mainly with respect to its role in directing eye movements. Visual input from the retina, or command input from the cerebral cortex, create a bump of activity in the tactile map, which, if strong enough, induces a saccadic eye movement. Even in primates, however, the superior colliculus is also involved in generating spatially directed head turns, arm-reaching movements, and shifts in attention that do not involve any overt movements. In other species, the tectum is involved in a wide range of responses, including whole-body turns in walking rats, swimming fishes, or flying birds, tongue strikes toward prey in frogs, fang strikes in snakes, etc. In some vertebrates, including fish and birds, the tectum is one of the largest components of the brain. In mammals, and especially primates, the massive expansion of the cerebral cortex reduces the tectum, superior colliculus, to a much smaller fraction of the whole brain. It remains nonetheless important in terms of function as the primary integrating center for eye movements. Note on terminology: this article follows terminology established in the literature, using the term superior colliculus when discussing mammals and optic tectum when discussing either specific non-mammalian species or vertebrates in general. Structure: the two superior colliculi sit below the thalamus and surround the pineal gland in the vertebrate midbrain. It comprises the dorsal aspect of the midbrain, posterior to the periaqueductal gray and immediately superior to the inferior colliculus. The inferior and superior colliculi are known collectively as the corpora quadrigemina, Latin, quadruplet bodies. The brachium of superior colliculus, or superior brachium, extends laterally from the superior colliculus, and, passing between the pulvinar and medial geniculate body, is partly continued into an eminence called the lateral geniculate body, and partly into the optic tract. Neural circuit: the microstructure of the optic tectum/superior colliculus varies across species. As a general rule, there is always a clear distinction between superficial layers, which receive input primarily from the visual system and show primarily visual responses, and deeper layers, which receive many types of input and project to numerous motor-related brain areas. The distinction between these two zones is so clear and consistent that some anatomists have suggested that they should be considered separate brain structures. In mammals, neuroanatomists conventionally identify 7 layers. The top 3 layers are called superficial. Lamina I or SZ, the stratum zonale, is a thin layer consisting of small myelinated axons together with marginal and horizontal cells. Lamina II or SGS, the stratum graum superficial gray layer, contains many neurons of various shapes and sizes. Lamina 3 or so, the stratum opticum, optic layer, consists mainly of axons coming from the optic tract. Next come two intermediate layers. Lamina 4 or SGI, the stratum graum intermedium, intermediate gray layer, is the thickest layer, and is filled with many neurons of many sizes. This layer is often as thick as all the other layers together. It is often subdivided into upper and lower parts. Lamina V or Psi, the stratum album intermedium, intermediate white layer, consists mainly of fibers from various sources. Finally come the two deep layers. Lamina 6 or SGP, the stratum graum profundum, deep gray layer, consists of loosely packed neurons and myelinated fibers. Lamina 7 or SAP, the stratum album profundum, deep white layer, lying directly above the periaqueductal gray, consists entirely of fibers. The superficial layers receive input mainly from the retina, vision-related areas of the cerebral cortex, and two tectal-related structures called the pretectum and parabigeminal nucleus. The retinal input encompasses the entire superficial zone, and is bilateral, although the contralateral portion is more extensive. The cortical input comes most heavily from the primary visual cortex, area 17, the secondary visual cortex, areas 18 and 19, and the frontal eye fields. The parabigeminal nucleus plays a very important role in tectal function that is described below. In contrast to the vision-dominated inputs to the superficial layers, the intermediate and deep layers receive inputs from a very diverse set of sensory and motor structures. Most areas of the cerebral cortex project to these layers, although the input from association areas tends to be heavier than the input from primary sensory or motor areas. However, the cortical areas involved, and the strength of their relative projections differs across species. Another important input comes from the substantia nigra, pars reticulata, a component of the basal ganglia. This projection uses the inhibitory neurotransmitter GABA, and is thought to exert a gating effect on the superior colliculus. The intermediate and deep layers also receive input from the spinal trigeminal nucleus, which conveys somatosensory information from the face, as well as the hypothalamus, zona incerta, thalamus, and inferior colliculus. In addition to their distinctive inputs, the superficial and deep zones of the superior colliculus also have distinctive outputs. One of the most important outputs goes to the pulvinar and lateral intermediate areas of the thalamus, which in turn project to areas of the cerebral cortex that are involved in controlling eye movements. There are also projections from the superficial zone to the pretectal nuclei, lateral geniculate nucleus of the thalamus, and the parabigeminal nucleus. The projections from the deeper layers are more extensive. There are two large descending pathways, traveling to the brainstem and spinal cord, and numerous ascending projections to a variety of sensory and motor centers, including several that are involved in generating eye movements. Mosaic structure. On detailed examination, the collicular layers are actually not smooth sheets, but divided into a honeycomb arrangement of discrete columns. The clearest indication of columnar structure comes from the cholinergic inputs arising from the parabigeminal nucleus, whose terminals form evenly spaced clusters that extend from top to bottom of the tectum. Several other neurochemical markers including calretinin, parvalbumin, GAP43, and NMDA receptors, and connections with numerous other brain structures in the brainstem and diencephalon, also show a corresponding inhomogeneity. The total number of columns has been estimated at around 100. The functional significance of this columnar architecture is not clear, but it is interesting that recent evidence has implicated the cholinergic inputs as part of a recurrent circuit producing winner-take-all dynamics within the tectum, as described in more detail below. All species that have been examined, including mammals and non-mammals, show compartmentalization, but there are some systematic differences in the details of the arrangement. In species with a streak-type retina, mainly species with laterally placed eyes, such as rabbits and deer. The compartments cover the full extent of the SC. In species with a centrally placed fovea, however, the compartmentalization breaks down in the front, rostral, part of the SC. This portion of the SC contains many fixation neurons that fire continually while the eyes remain fixed in a constant position. Related structures. The optic tectum is closely associated with an adjoining structure called nucleus ischiae, which has drawn great interest recently because of new evidence that it makes a very important contribution to tactile function. In mammals, where the term superior colliculus is generally used instead of optic tectum, this area is called the parabigeminal nucleus. Once again, this is simply a case of two different names being used for the same structure. The nucleus ischiae is divided into two parts, called pars manucellularis, IMC, the part with the large cells, and pars parvicellularis, IPC, the part with the small cells. As illustrated in the adjoining diagram, connections between the three areas tectum, IPC, and IMC are topographic. Neurons in the superficial layers of the tectum project to corresponding points in IPC and IMC. The projections to IPC are tightly focused, while the projections to IMC are somewhat more diffuse. IPC gives rise to tightly focused cholinergic projections both to IMC and the tectum. In the tectum, the cholinergic inputs from IPC ramify to give rise to terminals that extend across an entire column, from top to bottom. IMC, in contrast, gives rise to GABergic projections to IPC and tectum that spread very broadly in the lateral dimensions, encompassing most of the retinotopic map. Thus, the tectum IPC-IMC circuit causes tactile activity to produce recurrent feedback that involves tightly focused excitation of a small column of neighboring tactile neurons, together with global inhibition of distant tactile neurons. Function. The history of investigation of the optic tectum has been marked by several large shifts in opinion. Before about 1970, most studies involved non-mammals, fish, frogs, birds, that is, species in which the tectum is the dominant structure that receives input from the eyes. The general view then was that the tectum, in these species, is the main visual center in the non-mammalian brain, and, as a consequence, is involved in a wide variety of behaviors. From the 1970s to 1990s, however, neural recordings from mammals, mostly monkeys, focused primarily on the role of the superior colliculus in controlling eye movements. This line of investigation came to dominate the literature to such a degree that the majority opinion was that eye movement control is the only important function in mammals, a view still reflected in many current textbooks. In the late 1990s, however, experiments using animals whose heads were free to move showed clearly that the SC actually produces gaze shifts, usually composed of combined head and eye movements, rather than eye movements per SC. This discovery reawakened interest in the full breadth of functions of the superior colliculus, and led to studies of multisensory integration in a variety of species and situations. Nevertheless, the role of the SC in controlling eye movements is understood in much greater depth than any other function. Behavioral studies have shown that the SC is not needed for object recognition, but plays a critical role in the ability to direct behaviors toward specific objects, and can support this ability even in the absence of the cerebral cortex. Thus, cats with major damage to the visual cortex cannot recognize objects, but may still be able to follow and orient toward moving stimuli, although more slowly than usual. If one half of the SC is removed, however, the cats will circle constantly toward the site of the lesion, and orient compulsively toward objects located there, but fail to orient at all toward objects located in the opposite hemifield. These deficits diminish over time but never disappear. Eye movements. In primates, eye movements can be divided into several types, fixation, in which the eyes are directed toward a motionless object, with eye movements only to compensate for movements of the head, smooth pursuit, in which the eyes move steadily to track a moving object, saccades, in which the eyes move very rapidly from one location to another, and vergence, in which the eyes move simultaneously in opposite directions to obtain or maintain single binocular vision. The superior colliculus is involved in all of these, but its role in saccades has been studied most intensively. Each of the two colliculi, one on each side of the brain, contains a two-dimensional map representing half of the visual field. The fovea, the region of maximum sensitivity, is represented at the front edge of the map, and the periphery at the back edge. Eye movements are evoked by activity in the deep layers of the SC. During fixation, neurons near the front edge, the foveal zone, are tonically active. During smooth pursuit, neurons a small distance from the front edge are activated, leading to small eye movements. For saccades, neurons are activated in a region that represents the point to which the saccade will be directed. Just prior to a saccade, activity rapidly builds up at the target location and decreases in other parts of the SC. The coding is rather broad, so that for any given saccade the activity profile forms a hill that encompasses a substantial fraction of the collicular map. The location of the peak of this hill represents the saccade target. The SC encodes the target of a gaze shift, but it does not seem to specify the precise movements needed to get there. The decomposition of a gaze shift into head and eye movements and the precise trajectory of the eye during a saccade depend on integration of collicular and non-collicular signals by downstream motor areas, in ways that are not yet well understood. Regardless of how the movement is evoked or performed, the SC encodes it in retinotopic coordinates, that is, the location of the SC hill corresponds to a fixed location on the retina. This seems to contradict the observation that stimulation of a single point on the SC can result in different gaze shift directions, depending on initial eye orientation. However, it has been shown that this is because the retinal location of a stimulus is a nonlinear function of target location, eye orientation, and the spherical geometry of the eye. There has been some controversy about whether the SC merely commands eye movements, and leaves the execution to other structures, or whether it actively participates in the performance of a saccade. In 1991, Munoz et al., on the basis of data they collected, argued that, during a saccade, the hill of activity in the SC moves gradually, to reflect the changing offset of the eye from the target location while the saccade is progressing. At present, the predominant view is that, although the hill does shift slightly during a saccade, it does not shift in the steady and proportionate way that the moving hill hypothesis predicts. However, moving hills may play another role in the superior colliculus. More recent experiments have demonstrated a continuously moving hill of visual memory activity when the eyes move slowly while a separate saccade target is retained. The output from the motor sector of the SC goes to a set of midbrain and brainstem nuclei, which transform the place code used by the SC into the rate code used by oculomotor neurons. Eye movements are generated by 6 muscles, arranged in 3 orthogonally aligned pairs. Thus, at the level of the final common path, eye movements are encoded in essentially a Cartesian coordinate system. Although the SC receives a strong input directly from the retina, in primates it is largely under the control of the cerebral cortex, which contains several areas that are involved in determining eye movements. The frontal eye fields, a portion of the motor cortex, are involved in triggering intentional saccades, and an adjoining area, the supplementary eye fields, are involved in organizing groups of saccades into sequences. The parietal eye fields, farther back in the brain, are involved mainly in reflexive saccades, made in response to changes in the view. The SC only receives visual inputs in its superficial layers, whereas the deeper layers of the colliculus receive also auditory and somatosensory inputs and are connected to many sensorimotor areas of the brain. The colliculus as a whole is thought to help orient the head and eyes towards something seen and heard. The superior colliculus also receives auditory information from the inferior colliculus. This auditory information is integrated with the visual information already present to produce the ventriloquist effect. Distractibility. As well as being related to eye movements, the SC appears to have an important role to play in the circuitry underpinning distractibility. Heightened distractibility occurs in normal aging and is also a central feature in a number of medical conditions, including attention deficit hyperactivity disorder, ADHD. Research has shown that lesions to the SC in a number of species can result in heightened distractibility and, in humans, removing the inhibitory control on the SC from the prefrontal cortex, therefore increasing activity in the area, also increases distractibility. Research in an animal model of ADHD, the spontaneously hypertensive rat, also shows altered collicular-dependent behaviors and physiology. Furthermore, amphetamine, a mainstay treatment for ADHD, also suppresses activity in the colliculus in healthy animals. Other animals. Primates. It is usually accepted that the primate superior colliculus is unique among mammals, in that it does not contain a complete map of the visual field seen by the contralateral eye. Instead, like the visual cortex and lateral geniculate nucleus, each colliculus represents only the contralateral half of the visual field, up to the midline, and excludes a representation of the ipsilateral half. This functional characteristic is explained by the absence, in primates, of anatomical connections between the retinal ganglion cells in the temporal half of the retina and the contralateral superior colliculus. In other mammals, the retinal ganglion cells throughout the contralateral retina project to the contralateral colliculus. This distinction between primates and non-primates has been one of the key lines of evidence in support of the flying primates theory proposed by Australian neuroscientist Jack Pettigrew in 1986, after he discovered that flying foxes, megabats, resemble primates in terms of the pattern of anatomical connections between the retina and superior colliculus. Other vertebrates. The optic tectum is one of the fundamental components of the vertebrate brain, existing across the full range of species from hagfish to human. See the brain article for background. Some aspects of the structure are very consistent, including a structure composed of a number of layers, with a dense input from the optic tracts to the superficial layers and another strong input conveying somatosensory input to deeper layers. Other aspects are highly variable, such as the total number of layers, from 3 in the African lungfish to 15 in the goldfish, and the number of different types of cells, from 2 in the lungfish to 27 in the house sparrow. In hagfish, lamprey, and shark it is a relatively small structure, but in teleost fish it is greatly expanded, in some cases becoming the largest structure in the brain. See the adjoining drawing of a codfish brain. In amphibians, reptiles, and especially birds it is also a very significant component, but in mammals it is dwarfed by the massive expansion of the cerebral cortex. In snakes that can detect infrared radiation, such as pythons and pit vipers, the initial neural input is through the trigeminal nerve instead of the optic tract. The rest of the processing is similar to that of the visual sense and, thus, involves the optic tectum. Lamprey. The lamprey has been extensively studied because it has a relatively simple brain that is thought in many respects to reflect the brain structure of early vertebrate ancestors. Beginning in the 1970s, Sten Grill-Laner and his colleagues at the Karolinska Institute in Stockholm have used the lamprey as a model system to work out the fundamental principles of motor control in vertebrates, starting in the spinal cord and working upward into the brain. In a series of studies, they found that neural circuits within the spinal cord are capable of generating the rhythmic motor patterns that underlie swimming, that these circuits are controlled by specific locomotor areas in the brainstem and midbrain, and that these areas in turn are controlled by higher brain structures including the basal ganglia and tectum. In a study of the lamprey tectum published in 2007, they found that electrical stimulation could elicit eye movements, lateral bending movements, or swimming activity, and that the type, amplitude, and direction of movement varied as a function of the location within the tectum that was stimulated. These findings were interpreted as consistent with the idea that the tectum generates goal-directed locomotion in the lamprey as it does in other species. Bats. Bats are not, in fact, blind, but they depend much more on echolocation than vision for navigation and prey capture. They obtain information about the surrounding world by emitting sonar chirps and then listening for the echoes. Their brains are highly specialized for this process, and some of these specializations appear in the superior colliculus. In bats, the retinal projection occupies only a thin zone just beneath the surface, but there are extensive inputs from auditory areas, and outputs to motor areas capable of orienting the ears, head, or body. Echoes coming from different directions activate neurons at different locations in the collicular layers, and activation of collicular neurons influences the chirps that the bats emit. Thus, there is a strong case that the superior colliculus performs the same sorts of functions for the auditory guided behaviors of bats that it performs for the visual guided behaviors of other species. Bats are usually classified into two main groups: Microchiroptera, the most numerous, and commonly found throughout the world, and Megachiroptera, fruit bats, found in Asia, Africa, and Australasia. With one exception, megabats do not echolocate, and rely on a developed sense of vision to navigate. The visual receptive fields of neurons in the superior colliculus in these animals form a precise map of the retina, similar to that found in cats and primates.
Please subscribe and thanks for watching.